So I chose the topic of cancer because it's something that's obviously quite prevalent in the world, and it is something that we study both at GCSE, more at GCSE. We don't study it much at A level, so this is going a bit beyond the syllabus, but we do study what can cause cancer and mutations in DNA. So I thought I'd start with a couple of questions to get you thinking. The first one is a few statistics. So how many people in the Western world develop cancer? So we've got 1 in 2, 1 in 3, 1 in 5, or 1 in 6. And then the second question was, how many different types of cancer are there? And we've got 300, 200, 700, or 47. So the answer to the first one is actually 1 in 2. A generation ago, so when I was sort of in school age, it was 1 in 3. But that has increased quite rapidly in the last sort of 15 years. So 1 in 2 people will develop cancer. Obviously not everyone of that will die. Lots of people will get cured, but about 50% of people will suffer it at some point in their life. And the next one, there's about 200 different types of cancer that we know of at the moment. So there's quite a lot of different cancers. Some are extremely rare, and then obviously others are much more common. So the next question was, what is the most common cancer in the UK? So you've got breast cancer, lung cancer, colorectal cancer, or skin cancer. And then number 4 was, what is the biggest risk factor associated with developing cancer? So drinking alcohol, sun exposure, poor diet, or smoking. So for question number 3, the most common cancer in the UK is lung cancer. I think this is global as well. The most common cancer is lung cancer. And the biggest risk factor links to that is actually smoking. Obviously everyone associates smoking with lung cancer, but smoking causes a huge number of other cancers as well, or can cause. All of these are linked to some form of cancer. All of these can increase your risk of cancer, but the biggest one is smoking. And the last couple of questions, some cancers are genetic, true or false? And very old statistics, so this is 12 years old, but in 2002, how many cases of breast cancer were recorded in the USA? So 205,000, 30,800, 145,700, or 189,000. So the first one, cancer be genetic, it is true. It's a very small amount of cancers that are genetic, but there are a couple. The one you probably have heard of is breast cancer and the BRCA gene. It was made famous a few years ago by Angelina Jolie testing for that gene and having it. And the question 6, it was 205,000, but this is back in 2002, so it's probably increased since then. So what is cancer? Cancer is basically a cell in your body which has mutated, so the DNA has changed, and that causes it to divide uncontrollably. So it divides much, much quicker than a normal cell. So this is a human melanoma cell, so skin cell, undergoing cell division. So this is just as it's dividing into 2 different cells. The colours are caused by UV dyes that we've added to the cell to stain different components so that you can see them more easily. And this here, so this says interphase up here. This is the cell cycle. So this is something that is studied briefly at GCSE. And then at A-level, we study this in a lot of detail, what happens at each stage. So this is what happens in a normal cell. And this takes about 24 hours. So all your body cells are doing this, and it takes about 24 hours for a cell to divide in a human. Different organisms are faster or slower. So bacteria, for example, divide every 20 minutes, which is why they can make you sick so quickly. But our cells are much slower because there's a lot going on. They have to grow. So everything that's inside your cell needs to make a copy of itself, and your DNA needs to make a copy of itself as well. So this all takes time so that your two new cells will both be identical. Within this cell cycle, you've also got these little checkpoints. So this is where your cell sort of pauses for a moment and checks that everything is correct. So for example, here it can check that the DNA has replicated correctly. Up here it can check that all of the different cell components, so the organelles, like you may have heard of mitochondria, which make energy. So it's checking that those have copied themselves correctly. If something has gone wrong, the cell will stop and it will try and repair the mistake. And if it can't repair it, it will kill itself, which is something called apoptosis. So your cell is very good at checking for mistakes. However, sometimes it doesn't check it properly, and that's when cancer can develop. So this is what your cells do on a daily basis. They have something called contact inhibition, or sometimes called density dependent inhibition. So basically that means that they'll just grow in one layer. So here you can see the cells are starting to divide. And then here, when they all start touching each other, they stop. So the contact is touching each other. Inhibition means to stop. And then they'll just grow in one layer. If you take some cells out, then they'll start growing again because they're not in contact anymore. So then they'll start to grow again to fill that space. So they'll always just grow into one layer. In the lab, we can add chemicals called growth factors, which will then tell the cells to divide a bit more. And so then they can grow into more than one layer. We do this just for research, really, for different scientific experiments. So this shouldn't happen in your body. And this is how the growth factors work. So this is cell signaling, which is something we study in A-level. So the growth factor is on the outside of your cell, and then it binds to a protein, which is on your cell membrane, your plasma membrane. And then once it binds to the protein, you get all of these chemical changes inside the cell, and they will go into the nucleus, which is where your DNA is, and it will tell your cell to start doing division. It will tell your cell to start dividing. So cancer cells are a little bit different because they don't stop dividing, and they can divide much quicker than normal body cells. So this here would be a cancer cell, and it's about to undergo division, and then all around it are normal body cells. So cancer cells divide much quicker, and that's when they form a tumour or a growth. So this is just a picture of a breast cancer cell under a microscope. So traits of cancer cells, so how cancer cells behave. they are independent of growth signals from other cells. So they are not controlled by normal body signals. And cancer genes are often called oncogenes. They ignore stop signals. So in the cell cycle where you had those checkpoints, they can stop the cell cycle. But in cancer cells, they're not stopped. They just continue. So the DNA would not be repaired and the cell would divide even if it's faulty. So these are called tumor suppressor genes. So they suppress, they stop the cell from dividing. But if these genes are mutated, then they won't work and they won't stop the tumor. So here's an example, a very simple image from an A-level textbook. This is a tumor suppressor gene. So this is a normal gene in your DNA, and it makes a protein via a process called protein synthesis. So that's something which is studied briefly at GCSE and then in depth at A-level. And it will make a protein which then stops the cells from dividing. If the DNA mutates and changes, then it might not make the protein, or it will make the protein, but it's faulty and it's not correct, so it won't work. And that means that the cells are not stopped and they just keep dividing and dividing. And that's what would cause the tumor cell. Also, cancer cells, they don't do apoptosis, which is sometimes called cell suicide or programmed cell death. So the cells just keep dividing. And there's no limit to those cell divisions because they don't have that contact inhibition. So they just keep going and going and going. And then finally, they have to do something called angiogenesis, which means making new blood vessels, because the cells need a lot of energy to do the division. So they need oxygen and they need nutrients to be able to produce that energy. And that can only come from the blood. So they grow blood vessels around the tumor so they can get the oxygen and the nutrients. And then once they've done that, they're able to do metastasis. So you may have heard the word, or you may have heard the phrase, the cancer has metastasized, which means it's spread to other parts of the body. So a benign tumor does not move from where it grows. And often we say benign is non-cancerous, but malignant is very invasive, so it can invade other parts of the body. So once angiogenesis has happened, these cancer cells can get into the blood and then they can move to other parts of the body and then they can grow in another part of the body and form another cancer. So this was an example in breast cancer. So this is a reason why it's very, very important to catch cancer early. Because if you catch it early, it's very treatable and you don't want it to spread to other parts of the body. So here, this would be an early breast cancer where you can see a very small tumor starting to grow. If you catch it at this point, it's very simple to treat. They can just cut out the tumor and then you'd normally have some kind of chemotherapy or radiotherapy just to kill any cells to make sure you haven't left any cancer cells there. If you don't find it early, you can see the tumor will start to grow and that they're starting to invade other tissues. At the moment, it's still only breast tissue, but it's just getting bigger. And then here you've got lymph vessels. So these are part of your lymphatic system, which is part of your immune system. If the cancer keeps growing, the cancer cells can break off the tumor and they can get into this lymphatic system. And once that's happened, the cancer cells will move to other parts of the body, and then they can grow new tumours somewhere else. Once you get to this point, it's much more difficult to treat cancer. So you want to catch it very early. This you may have seen before. So this is a picture of chromosomes. So in human cells, we have 46 chromosomes, or as you can see here, 23 pairs. So 1, 2, 3. The 23rd one is the sex chromosome. So if you're female, you'll be XX, and if you're male, you'll have an X and a Y. But in humans, all of our chromosomes are in pairs. So you get one from your mum and one from your dad. What happens in cancer is these chromosomes, they're made of DNA, so they have genes on them, but the DNA may mutate, so it changes. So it can change the DNA sequence. Some of these mutations are known as germline, which means they happen in the gametes, which are the sex cells, so the egg or the sperm. And if that happens, then it does get passed on to the children. But most cancers are somatic. So a somatic cell is just a body cell, a normal body cell. So not sperm or egg, but any other type of cell in your body. So skin, muscle, not that you get many muscle cancers. So the process is called oncogenesis because it's making an oncogene, which is the cancer gene. So it can be somatic, which is a normal body cell, or inherited, so it comes from your parents. If you inherit it, it means every cell in your body has that mutation, so you've got a higher risk of getting cancer. It doesn't mean you definitely will get cancer, it just increases your risk a little bit. Somatic mutations are ones that happen throughout your lifetime. So these ones you don't pass on to your children because they happen in your body cells. So this picture here gives you an idea of the main causes of cancer are somatic. There's very few that are inherited. There's some which are a bit of both. So I've given the example of the BRCA gene here. So this is the one that increases your risk of breast cancer, but also sort of cervical and womb cancer as well. But only about 5 to 10% of these cancers have a hereditary component. So in the UK, you can get tested now to see if you have this gene. But you can only get tested on the National Health Service if there's a history of breast cancer in your family. Then you can get tested for free to see if you have this gene. But even if you have the gene, it does not mean you will get cancer. But it does increase your risk by about 70%. So it's quite a difficult decision to know. Do you want to know if you've got the gene, or do you not want to know? So it's very much your choice if you get tested or not. So two types of cancer genes: Tumour suppressor, so they're the ones that stop the cancer, and then oncogene is the gene that causes cancers. So we know of about 400 genes at the moment through gene sequencing, human genome sequencing. There are more to be discovered. We just obviously haven't sequenced all of them yet. So this one is your tumour suppressor gene, TS. So the genes are carried on chromosomes. So this light blue colour here is the gene that makes the tumour suppressor protein. If that gets mutated, like here, that means that this gene will not stop the cell cycle. So therefore, you can get cancer. The other one is oncogenes. So an example is RAS, but there's lots of different examples. These actually promote cell growth and division. So they tell the cell to divide. Again, if you mutate it, it means that the cell will just divide quicker and uncontrollably, and that can lead to cancer as well. So mutations are the main cause of cancer. So mutations are changes in the DNA. One mutation is unlikely to cause cancer. You have to have more than one mutation. So it's an accumulation of lots of mutations, which is why more people get cancer at an older age than younger people, because the older you get, the more mutations you have in your DNA, just because you've had more exposure to environmental factors that can damage your DNA, and your cells they're not quite as good at repairing. The older you get, they sort of slow down as well. So yeah, mutation is a change in the DNA. It can be caused by UV light from the sun, X-rays. So if you've ever had an X-ray in a hospital, you'll notice that the radiographer goes behind a LED screen, and they often wear the vests, and that's to stop them being exposed to the X-ray. Natural or synthetic chemicals, so chemicals in the air, pollution from cars, asbestos was a big one a few years ago, but also viruses. So HPV, human papilloma virus, has been linked to increasing the risk of cervical cancer. So in the UK, about 12 years ago, they developed a vaccine for HPV. So all girls get the vaccine for HPV in the UK. They've just released one for boys as well. But at the moment, it's still girls that get it. And it got known as the sort of cancer vaccine, which is not true, because it does not stop you getting cancer. It stops you getting the virus, which could cause cancer. So it's a bit of a misbelief there. So these are the different types of mutations. So these are your chromosomes here. So you could have a deletion. So you can see here, this chromosome has changed because a whole gene has been deleted. You can get duplication. So homologous chromosome means, homo means the same. So it means the pair. So you've got the one from your mum and the one from your dad. So when your DNA replicates or copies itself, sometimes it makes a mistake, and you can see here that the gene from this chromosome has been added to this one, so it's made it longer. Inversion is just where the DNA swaps places within the chromosome. And reciprocal translocation happens between different chromosomes. So it could be between chromosome number 1 and chromosome number 4, for example. And in this example, they've swapped DNA. So this one is the reciprocal one. So chromosome number 9 has lost a gene, and it's been added to chromosome number 22. And when this happens, you make a cancer gene. So it's known as the Philadelphia chromosome. It's the one that causes chronic myeloid leukemia. So that cancer is caused by reciprocal translocation. So again, a few examples here. This is something we do at A-level, so sort of identifying different mutations. So you've got a sequence of DNA here. So all of your DNA is made up of 4 different chemicals called bases. So you've got A, which is adenine, C, cytosine, G, guanine, and T, thymine. So at GCSE, you will learn that and how they pair together. And then at A level, you'll learn about how the DNA copies itself. So if you look at sequence, all sequence number ones are all the same. And then sequence number 2 is where the mutation has happened. So if you compare number 1 here with number 2 here, you might notice ACTC is the same. And then the next one here is a C, but in this one it's a G. So this mutation is called a substitution because what's happened is the G has been changed to a C. So the DNA sequence has changed a little bit. The next one's a bit easier to spot. You can see that this is much shorter. So some of the bases have been removed. So we just call it a deletion. The next one, if you compare it, you can see this one's a little bit longer than this one. So it's had some bases added to it. So sometimes we call it addition because it maybe is just added one. This one we're calling insertion because it's added more than one. The next one, if you compare them, they're almost the same. This one's inversion. So some of the bases have just swapped places. So the length is the same, but the letters have swapped places. And then the last one is longer again. And this one is called duplication because it's got the same bases. It's just repeated some of them twice. So there's lots of different types of mutations there. Anyone who's studied biology before, there are more. You might have heard of frame shifts. So the mutation causes a frame shift. Again, that's something we study at A level. So cancer is when your cells divide uncontrollably, as we've said. Normally, your cells have to divide, and they divide to replace dead cells in your body. So your skin, for example, the top layer of your skin is actually dead and is falling off all the time. So most of the dust in your home is your dead skin cells. So underneath, your skin is making new cells, and that's happening all the time. Your liver is a great example as well. Your liver is the only organ that can regenerate itself. So if you chopped some of your liver off, or if you cut it in half, it would grow back to its normal size. No other part of your body will do that, so don't try it. Also for growth, so for younger people, if you're still growing taller, for children especially, you're doing a lot of cell division because you're getting taller. Once you get to adults, our cells are not dividing as much because we're not growing taller anymore. So we tend to just be doing repair. But cancer cells just divide uncontrollably. They just don't stop dividing. And that can lead to problems because they can destroy normal, healthy cells and organs. So the mutation that we looked at causes the cancer cells. So they're called oncogenes. It's thought that several mutations need to occur to give you cancer. So like I said, one mutation will probably be okay. Also, one mutation, your cell is very good at correcting it. So it's when you get lots of them at the same time. So they don't kill themselves. So normally if you get a mutation, your cell will self-destruct. But if it's a cancer cell, it won't, and it will just keep dividing, producing millions and millions of new cells. So remember, benign tumours are they're often called non-cancerous. So they don't spread to other parts of the body, but they can cause problems. So for example, if you get a brain tumour, even if it's not cancerous, it can grow and put pressure on the brain, which can obviously cause problems. So that one you would probably have to have removed. But warts, they're like little growths on your skin. They're actually just where your skin cells divide too much. And they're benign tumours, and they don't cause any harm at all. You can leave them. People often have them removed because they don't like the look of them, but they're not harmful at all. Malignant tumours are the ones that spread and can cause secondary tumours, and that's the one that's called metastasis. And they're bad because they can prevent your body from functioning normally. They can block, especially tumours, they can block blood vessels as well, stop blood getting to certain parts of the body. And they require lots of energy as well. So they take lots of oxygen and nutrients from your blood so that they can divide, and that can make you quite tired. So the last couple of slides just sort of show you this in images. So this is your DNA structure. It's called a double helix. You've probably seen this before. And those sequences, the A, the T, the G, and the C, are paired up along the middle here. So normally your cells will copy this DNA and they'll just make new cells, and the two new cells will be identical. But if one of these gets mutated, so this base here, the A, T, G, C, if they change, that will cause a mutation, and it means that the new cell will not be the same as the original cell, and that could then cause cancer. And that mutation can be caused by radiation or chemicals. So this cell here has had one mutation, but all of the other ones are still normal. So not a lot has happened yet. You can then, so this cell that's mutated is going to then divide and spread like this. But then another cell could get a second mutation. So at the moment, these mutated cells aren't causing any problems. They're not cancerous yet. But this one has now mutated again. And that mutated cell is starting to grow into a tumour. And then you can see this cell has mutated a third time. So it's changed even more. And the change might be that it grows quicker, it divides quicker. So that can cause the growth of the tumour. The blood vessels down here are still in their normal position. They haven't moved. We then get, so after the third mutation, you start to get the growth of this tumour. And then after the fourth mutation, it's causing a more aggressive cancer. So you can see here, it's starting to break through the membrane. At the moment, the tumour is still in one place, but this fourth mutation is going to cause more problems because it's starting to break through. And if it can break through the membrane and get into the blood, it then means it's going to metastasize and spread to other parts of the body. So at this point, it's starting to break through the membrane. So that means it's going to spread to other parts of the body. And when it does that, this is the angiogenesis phase. So the blood vessels start to grow around the tumour so that the tumour can get more oxygen, more nutrients, so that the cells can keep dividing. And also these cancer cells can now get into the blood. And once they're in the blood, like here, they can spread to other parts of the body. So they'll move around in the blood. Interestingly, cancer cells can't survive very well in the blood, but you've got hundreds and thousands of them moving in the blood. So that increases their chance of survival. So once they get into the blood, they're going to spread to another part of the body. And they then need to attach to the blood vessel and get out of the blood vessel and start growing in another part of the body. And that would then be a secondary tumour. And then the secondary tumour will start to grow, and then the same process can happen again. It will grow a new tumour, then it will do the angiogenesis, and then it could spread again. So the process starts all over again. Once you get to this stage of cancer, unfortunately, you're unlikely to survive. Once it's spread to another part of the body, it's very hard to treat. So this image just showed you the same thing happen again. So at the moment, there's some mutations. This is in the intestine, and you're starting to grow a small little tumour. At the moment, it's benign because it's in one place. It's not caused cancer yet. It's not moved anywhere. You then get more mutations, and it's known as in situ cancer because it's inside the body. It then mutates again and becomes invasive because it's going to get through the membrane of the small intestine, and it's going to get into your blood. And then once it's in the blood, it metastasizes and spreads to other parts of the body. So just to summarize, what causes these mutations is, or can be, all of these increase the risk. So UV radiation from the sun, difficult one because you have to have sunlight. Your body uses sunlight to make vitamin D, which is essential to your body. So you do need a small amount of sunlight. Depending on your skin color, someone with my skin color needs a minimum of sort of 15 minutes a day. So not long at all. If you've got darker skin color, you need slightly longer because your skin is darker, so it doesn't absorb as much. But you've got UVA and UVB radiation. So UVA is the longer wavelength, and that's the one, it does damage your DNA, but more in that it will cause your skin to age. So it causes wrinkles. Whereas UVB is the shorter wavelength, and that's the one that causes sunburn. And that's the one that's more dangerous and can lead to cancer. So when you're wearing suntan lotion, you've got to make sure it's got UVA and UVB protection. Smoking, like I said, there's over 300 different chemicals in a cigarette. So that causes cancer. Diet, and I've got a guy eating a burger here. It's not just burgers. When we say lifestyle, it is diet. It's, you know, one burger every now and again is not going to kill you, but having a very high fat diet or a very unhealthy diet, an unbalanced diet can increase the risk of cancer. But it's also things like how much exercise you get and drinking alcohol, for example, and stress as well. And then the last one was viruses. So this was a picture of cervical cancer. So these are the cells that have mutated in the epithelial cells in the cervix. So like I said, we've got a vaccine for it now, for the virus, not for the cancer. So yeah, that was it for the presentation. So yeah, if anyone has any questions.
Thank you, Nadine. John here.
Yeah.
Yeah, thank you very much. That was a really interesting and insightful presentation. We do have a few questions that people have posted, so I'll put those to you. But if anyone does have any more questions, please post them now. A couple of people have asked if we are sending the video, so we will send you a link to the video of the session afterwards. So don't worry about that. I'll put the actual questions on the content you presented to you now, and bear with me 1 second because we've got a few. Let's see if I can find the first one and do them in order. So Megan asked, how do you know if a tumour exists in the breast?
Oh, good question. So you're supposed to when they're really small, you can't, because often especially with breast cancer, it doesn't tend to cause much pain. So when they're really small, also they're really hard to detect. So they do recommend that you check every month. So you should literally just feel each. It's the same for men actually with testicular cancer. You're supposed to check every month. You're supposed to just feel around each breast, but also under the armpit, because that's where you've got lymph nodes. And if you feel any kind of little bump that you didn't notice before, you should always go and see your doctor. It might not be cancer. It could just be like a little thing called a lipoma, which is a growth of fatty tissue. But it's always better to get it checked. So they do recommend checking every month and just feeling.
Thank you, Nadine, and also, you know, from a UK perspective, there are huge public health campaigns around sort of checking for cancer as well, in the ways that she's described. Daniel has asked, is it possible to predict whether we know if a deletion, duplication, substitution, or inversion will occur before it actually takes effect? I hope I've said that right.
Yeah, no, that makes sense. I don't think so, no, because they're random. So all mutations are completely random. So we can't predict when they'll happen or where they'll happen or in what gene they'll happen in. The only thing we can sort of say is if you get tested for a certain gene that we've identified as increasing the risk of cancer, we can say that, well, you have got an increased risk of getting it, but we still can't say when it will happen. You know, we can't say, well, when you're 35, this mutation will take place and you will get cancer. And like I said, you might not even get it. So unfortunately, no.
Okay, thank you, Nadine. Daniel's also asked another question after that. Are we able to maintain and control the rate of the division of cancer cells, either benign or malignant? And if we are, can we use this to our advantage?
That is a really good question, actually. And most cancer drugs, that's exactly what they do. And there's a drug called, you've probably heard of it, thalidomide, which was made very famous in I think it was the early 70s, because it was given as a morning sickness drug for pregnant women. And then that caused birth abnormalities because it prevented cell division. And so babies were born with the very short arms and legs. So that drug's been looked at as a treatment for cancer because it prevents cells from dividing. But as opposed to sort of preventing it before it's happened, I don't believe so, no.
Okay, thanks, Nadine. Winnie's asked, are there any specific types of nutrients that expedite the growth of cancer cells?
There's always a lot of chat about antioxidants, isn't there? So there's something in your body called a free radical, which you can't stop free radicals. It's impossible. So you get them just from the environment anyway, but your body actually produces them normally during respiration. So when your mitochondria make energy, they do produce free radicals. And unfortunately, they do damage your DNA. But your cells normally can repair that damage. But there are certain antioxidants which are found in certain foods, especially fruits, which mop up or sort of help remove these free radicals. So that is supposed to prevent, I suppose, more damage to your DNA, which could potentially reduce the risk of cancer.
And I suppose the science is constantly evolving on this as well, because I know from personal experience, you're often hearing that certain types of foods can either increase or decrease your risk of cancer, or sometimes one type of food that is considered to be decreasing your risk of cancer, then they just someone else decides that it increases the risk.
It's always changing. It's like with butter. A few years ago, butter was bad. You should never eat butter. And now they're saying butter's better for you than margarine. So
there's always back and forth. Megan has asked, is it true to say that morning sunlight is better than afternoon sunlight?
Oh, do you know, this is a really interesting question, and you're probably not going to like me because I don't think I can answer it. Because we've always said stay out of the midday sun. So between like 11 and 3 o'clock is when the sun is highest in the sky, and that's when it's most damaging. And that's what we've always been told. And so like morning or evening is better because it's not as strong. But I was reading something recently in a research paper that was saying that yes, the afternoon sun is stronger and you shouldn't be out in it for too long, but from a health benefit it's actually better because your body absorbs the wavelengths it needs to make the vitamin D, whereas in the evening you get a different wavelength. I don't know about morning, but in the evening it said the wavelength is different, and that's you absorb more of the cancer causing wavelength in the evening. But don't quote me on that. It's just something I read recently in a research paper, so which I found quite interesting.
It's also a very interesting kind of message, isn't it? Because like you say, you know, people may protect themselves more in the mid-afternoon sun, and then decide in the late afternoon that they don't need to protect themselves as much as well.
Yep.
So the risk factor, I guess, can go up in that sense.
Exactly. Yeah.
Brilliant. Daniel's just asked a quick question that's just popped through. When and where do cancer cells tend to metastasize? I think I've said that right. I hope I said that right.
Yeah, no, you have. I don't know if I can answer that because it varies between patients. So it's not always the same for each patient, and some cancers are more likely to metastasize than others just because of where they grow in the body. So which is, I always mention breast cancer, but it's really important to catch that one early because you've got so many lymph nodes up around your chest area. It's easier for the cancer cells to get into the lymphatic system and spread, and the same with lung cancer, but other cancers are less likely to spread. So like skin cancer is a very easy one to treat and it's an easy one to spot, so it's less likely to metastasize because people catch it early. So it really depends at what stage you catch it, unfortunately. I hope that helps.
Thanks, Nadine. Winnie just mentioned that the mic is very soft. Apologies for that. It may be because we had both our mics on at the same time. I think that's all the questions that we've had come through. If anyone does want to post any final ones, please do now. But we'll presume that that's all your questions answered. Obviously, when we send you a copy of Nadine's slides and the video, please do ask any questions that come up, and we'll put them to either Nadine or one of her colleagues in the biology department to get back to you on. We also have another session taking place next week, which is kind of related to biology, and we're talking about how we help students prepare for medical schools. So that's a session that's going to be conducted next week with Dr. Julian Davies, who's the principal of Abbey College Cambridge and a doctor of biology, and Liz Elam, who's the principal of Abbey College Manchester. So please do check that out. You can find it on the website. And if you'd like to attend, please do. That would be very useful for anyone who's interested in studying biology and becoming a doctor. We don't have any more questions, so I think I'll let you say goodbye, Nadine, and then I'll finish off.
Thank you very much.
Yeah, thank you very much, guys. I hope you enjoyed the session, and we'll see you soon. Bye-bye.